Mesdames et messieurs, good evening, ladies and gentlemen. It's a real pleasure. My name is Michel, I'm the director of the Goodman Cancer Centre in the old ways. We're now the Goodman Cancer Centre for the past year due to the involvement of Maurice and Rosalind Goodman at the Cancer Centre. And we'll have quite a lot to tell you about tonight, particularly about the centre a little bit, but also on the one of the 4 evenings that we want to organize. So I will tell you a bit about the Cancer Centre in a few slides, not to take too much of the time, because we came here to learn about, to have a nice evening, to learn about cancer research, cancer, several issues, understand a bit what is the issues that clinicians and basic scientists have to deal with to bring to the public information. On top of that, we will talk about issues on prevention. Especially tonight, there's several things we'll talk about on how cancer develops. And we will look at this particular initial stage of cancer. Donc ce soir, on va surtout parler de l'initiation du cancer, comment un cancer se développe, quels sont les facteurs qui nous amènent à amener en fait un risque plus élevé dans le cancer. Donc ce soir, on va parler un peu des deux. Et au long de la soirée, vous allez voir différentes étapes de cette recherche. I just want to tell you a little bit very briefly what the Cancer Centre came from. First of all, it was established in 1978, quite a long time ago. It's not, however, the best, or not the best, the first cancer center in Quebec. L'Institut du Cancer de Montréal was established in the 1940s. And through the years, in fact, McGill here established, with the discovery of CEA by Phil Gold, the McGill Cancer Centre, which came later on to become the Goodman Cancer Centre. So if you, I don't think I have a pointer with me. Do you have a pointer, Sophie? No? Yeah. Oh. Next to the big screen. Ah, thank you. Thank you. So initially, the Cancer Centre was established by Phil Gold, as I mentioned to you, with one single discovery, which is still today one of the most important discoveries in cancer diagnostic, which is this marker called CEA. It's used still today, over 30 years later, as a marker around the world in cancer. I must say that Phil, this marker probably has made billions of dollars for company. At those days, Phil had to sell it for or set it to not too much for companies. This is not the same thing today. People work many, many years to develop these markers, and they are very precious for the people that develop it. Nevertheless, Phil Gold has given this to the community, and this is how this Cancer Centre started in those days. Many years later, there was a group established that the Cancer Centre also helped to do, which was the Molecular Oncology Group, which was located at the Royal Victoria Hospital. And they were doing very similar work at the hospital than what we're doing in the McGill Cancer Centre. And early in the 2000, when I became director, we fused these two groups together to become the McGill Cancer Centre FRSQ groups, long thing to say. But this group continued to thrive until we built the Pavillon Cancer ici at McGill. And eventually, last year, with the involvement of Rosalind and Maurice, this became one of the biggest centers in Canada in cancer research, which is the Rosalind and Maurice Cancer Research Centre, that you are just beside this room here. So I mentioned Phil. I think I just want to tell you a few of the discovery that came from here, because the reason where we basic scientists are mixing up with, in fact, clinicians, is to bring this knowledge of cancer research, cancer development of the disease, and our role is really to not only understand, but bring the tools that oncologists will use in their clinic. And so this is this process we're going to talk tonight, and I will go to each of the speakers, and tell you a bit what they're going to talk ce soir. So, je vous ai mentionné Phil Gold et le marqueur carcinoma brain antigen, CEA. It became now a kit that is sold around the world to detect cancer progressions, cancer treatment, and recurrence of the disease in many different ovarian cancer and breast cancers and others. But there's many other discovery in this center that occurs in the past and from different scientists. So again, one is the Wilms tumor gene. This is a gene involved in pediatric cancer, Wilms cancer, which is a kidney cancer in young children, which was discovered here and now is identified and treated around the world. P-glycoprotein is a protein that involves in cancer resistance. When you treat patients with various chemotherapy, very early on, people realized, and oncologists realized, there was a resistance to this treatment. And in fact, Philippe Gros uncovered that this protein was kind of a shuttling mechanism. As soon as the drug was coming in the cancer cells, this protein was pulling it out. So it was impossible to treat these resistant tumors. So again, just to give you a few examples, probably one of the latest one that I would like to tackle, Batoclax, is a drug that is now in clinic for lung cancer, which was developed here at the Cancer Center in Geminex, a company that started from the Cancer Center here. So all this to say that our issues is not only to understand the disease, but also to translate this in drugs and factors that can help clinicians and patients in the treatment of the disease. So at the Goodman Cancer Center, we have 5 units, 5 teams. And this is important because the research, as you know, is extremely expensive, very difficult, complex. So we put people with a different expertise together. And we have a group on breast cancer, which is associated also with the MUHC, a group in metabolism in cancer. Our second evening deals exactly with this. This group tried to understand, we hear a lot about broccoli and good health and eating well. As you know, we see this all the time. So this is very fine. But why is your broccoli or losing a bit of weight, which I'm trying to do badly, but why is this important for your cancer outcome? So we want to go beyond the broccoli. We want to understand this at the level of the cancer cells, try to see how the synergy we eat, the sugar, the fat, is transferred into energy and used differently by cancer cells versus normal cells. And this will be the second evening we have on our approach. We have a group on stem cells, en cellules souches. Et c'est important, ça, pour survivorship. Many cancer patients, plusieurs patients qui ont le cancer, vont être guéris, ils vont être en rémission. En cancer du sein, c'est très courant. You know, you have a cancer, you're treated, you have your 5 years remissions, and some of them rediscover cancer about 20 years afterwards, and then it restarts. Why is this? And so the research, we have to address this issue. Why is the fact that cancer cells, which are supposed to be gone, are there 25 years later? So this is a very difficult to understand, and we have to look at these cancer stem cells, how they hide, how can we recognize them, and we can treat them. So if we can pull them out of these tissues, perhaps we can design specific drugs against them. So that's one of the topics of the Goodman Cancer Center scientists. We have a group on development and epigenetics, and Jinzhao Yang, Dr. Yang will tell you a bit about epigenetics. A group also that looks at development. So, you know, one of the most beautiful example of development and cell growth is a baby. And José Dosti tonight will tell, we'll show probably a picture of her daughter. But to show you we grow well, but at one point we stop. And an adult will still need to have cells growing and replacing some of our tissue. But during that period of time, during that process, sometimes cells go awry and they develop cancer. So there's there's an important part of understanding how an embryo develops and which genes are involved, and those genes will come back again, not now to make a nice adult, a nice baby, but to become critical tumor genes in cancer. So that group at the Cancer Center studied development, epigenetic, and cancer. And finally, and it's really mainly the topic of tonight, DNA damage. So we talk a lot about environment, how we have mutation caused by the different carcinogen out there, the TV, the radon in our houses. So this group looks really at the mechanism of cancer mutations and how these single mutations can cause cancer later on, immediately or as we talked about 20 years later. So this is what we do. We have a team approach in the Cancer Center. Each of these teams work with a different affiliated hospital at McGill and different clinical group at the MUHC and also the Jewish General Hospital. So we're very much in partner, but at a different type of cancer. I am almost done with this. I know you want to listen to this topic tonight. So just to tell you that in cancer research, we just really, in fact, looking from the patient point of view where we have tumors and tissue. Many of the patients are giving their tissue to research, but we are the ones getting those in tumor banks. Our scientists look at the tumor banks, try to identify what in these tumors that we get from patients are important for their disease. And remember that eventually we'll go to personalized medicine, where we will not have thousands of patients for clinical trials, but each one of us will become its own clinical trial. So the future in the next 10 to 15 years is each of you will go to the hospital. Hopefully you will not, what I'm saying. But if a patient goes to the hospital, you will be able to treat it with different compounds to see which one works the best for you. And then you will be going home with the best treatment. And not one of 1,000 patients that hopefully you'll be the few in the cohort which has the most benefit. We would like every single one to have this benefit. So not only we identified those genes, we identified also how they work and how we can modulate them in tumors. So how we can develop new drugs and give few ideas to the pharma industry to where to go and to provide these treatment to our colleagues at the clinic. So I've done my speech about the Cancer Center, and if we want to come back to that later on in the questions, we'll do that. So the 3 sessions we'll have, the 3 other ones. Tonight we'll talk mainly about how the cancer cells initiate. How do we get this event that brings us cancer, and what can we do to prevent this? So there's few things we'll go tonight. The second session I mentioned to you sera sur how fuel cancer growth. How the cells in cancers use energy to develop. So we'll talk about that in the next evening. The third one is very important. Cancer metastasis. La plupart des cancers, s'ils ne faisaient pas de métastases, on pourrait survivre très bien. C'est le processus de métastase, en fait, qui est le plus dangereux et le plus létal pour un patient. Donc, we have, at the Cancer Center, several people. And in fact, it's a very nice group that started here and with scientists and clinicians at the MUHC that we have a very nice dynamic group on cancer metastasis. And we will talk about that, how it goes, how we can treat it, etc. in the session 3. And finally, I think we talked about survivorship. More and more, and this is the positive message, people are surviving cancer. And there's a real issues are just living with this. Are we going to get it back? Is it coming back in the next year or 2? So again, people with cancer will come and tell us about this. We'll have also clinicians that deal with this issue every day and point out a few suggestions on how we go, how we're followed, and making sure that this does not happen. And if it happens, we can treat it as fast as possible. And eventually, what we all want to do is that disease becomes, we will have no fear of that disease. It will be a chronic disease like diabetes, hopefully, in the good circumstances, where we can live with our disease for the rest of our life. So that's the best, I think, that will come. So that will be the 4th evening. So just before that, so tonight we'll have 5 speakers. As I mentioned, we'll talk about cancer initiation. We will have each of them, they're not long, about 5 or 6 slides each. We will also, after that, ask you if anybody has questions. En français ou en anglais. Jing Zhao is English is excellent. Is Chinese better? Is French, it could improve a bit. So I will help him if that's the case. But you're welcome pour demander en français ou en anglais, c'est pas un problème. Et puis on aura quelques questions. Mais à la fin de la soirée, on aura un panel en avant de tous les speakers où on pourra, à ce moment-là, soulever d'autres questions que vous avez au niveau du traitement, au niveau de la maladie, et on pourra répondre à ça. Donc, José will start talking to you about how the first mutation arises. So I'll tell you a bit about a background about genes, that we don't want to go too heavy here. It's supposed to be a pleasant evening, not an evening that you have to break your head. But we will talk to you a bit how this mutation arise, and that's what she will tell you. Jingjia will tell you, he's an expert on what we call epigenetics. Epigenetics is something that people did not believe before, but it's really the fact that genes, you can have cancer without necessarily having mutations. The environment changed the way your genes are expressed. So he will give you a good example of how, in this is a case, the prevention could be very important. Arne Posey will give you a bit more the aspect of research, how long it takes to get a new drugs, a new discovery. How do you identify a gene which is important, how we can target it, and give you some example of that happens in real life and why it takes so long sometimes. And I understand that public sometimes are quite impatient of having all this 30 years of cancer research, and we're still doing better. 60% of cancer are all cured now, but still it's not enough. So we will have a small break, which if I stop talking, and where after that we come back with Dr. Tonin. Pat was part of the international team that discovered BRCA1, a gene involved in hereditary breast cancer. This was a really big discovery, and she was the key person in the group here at McGill University that contributed to this. And they have the most happy person of the five, it looks like. Dr. Denis Cournoyer is a clinician, hemato-oncologist at the MUHC, which seems to be happy to answer all your clinical questions later tonight. But he has himself the ideas. He will be summarizing this part of the evening. So we'll finish with Denis, and we'll have questions after that. So hopefully this sounds a good evening, and I've talked already enough. I'll do the thank you later on. So I will stop there. Denis, if you want to, not Denis, but we need to rechange José the computer. During that time, I will just, you have a small questionnaire that you're welcome to answer. I think you're supposed to have that. Just to give us an idea if that's what, if we're answering the right question to you, because especially for those that, really would like to follow all the evenings, we must really improve on things. If there's questions that we've not answered, we'd like to do that in the second evening. If there are some things we want to change a little bit, we'll do it with pleasure. So. During the time they do this, I will. So this is this, and I'll take the microphone out. Yep.
Figure out how this. Okay, no, I think it's fine. Yes. Thank you. Yep, thank you. All right, good evening, everybody. Thank you all for coming tonight. This is my first time that I'm giving this type of lecture, so your feedback is going to be very much appreciated afterwards. I can adjust like this. This is better. My students tell me the same. I'm a low talker, apparently. So, okay, I'm going to try to scream out loud.
No, I think it's because I was wearing it wrong. If you turn it around, I think it'll look better.
It will look better.
It's just me that it not only look better, but be better. There you go.
Comme ça? Oh,
yeah.
Mon Dieu. Ah, là, je vais crier.
It's because I had the neck,
Okay, thank you very much. All right. So thank you also for the introduction, Michel. So my name is José, and I'm an assistant professor here at McGill, and my lab is also affiliated with the Goodman Cancer Research Centre. The type of research that I do in my lab is very basic. I'm actually relying always on clinician to keep me in check. Actually, that's why also I was asked to talk about the origin of cancer, because I mainly work with the genome. So the title of my talk today is The Origins of Cancer, because my goal today is really to tell you about the underlying causes of cancer. I'm going to be telling you a little bit about the terms that we use to discuss what cancer is. So I will introduce a lot of terminology. I will talk about what is the genome, what is a gene, what is a mutation, and what do we need, what do we mean by cancer susceptibility, and also what are the environmental factors that can lead to the development of cancer. So in order to understand what cancer is, we need to understand what normal growth is. And this is what Michel was talking about. This is why we have researchers studying the development of the human body and just general cell growth. So this is the first time that I use my daughter to explain something, but it's a very good occasion to brag a little bit. So my daughter was born January 1st, 2009. So after 28 hours of very hard labor, here she is a few minutes after she was born. And this is at Halloween, some 10 months later. So you can see that within 10 months, she's grown quite a bit. She was born at 7.5 pounds. She's now 21-some pounds. So she's tripled her body weight, and her muscles are really getting strong. She's crawling everywhere, and she's trying to walk. So I'm showing you this because I really want to illustrate the fantastic ability for the human body to grow. So in a span of 10 months, she tripled her body weight. In adults, we need to maintain this ability to grow because we need to change our skin. If we cut ourselves, we need to fix the scars. Our blood cells are always regenerating. So we need to, during adulthood, maintain this ability to replicate cells. And this process, however, in adults must be really tightly controlled. And when cells start dividing aberrantly, this is when cancer can develop. When we lose this control in the replication of cells, this is when you can develop masses, etc. So how do cells lose control? There's three main ways by which a cell can somehow all of a sudden decide that it's going to replicate aberrantly. So the first way is by acquiring mutations in the genome. So this is what I'm going to be talking about today mostly. So these changes there are changes in the letters of the genome. The other way that you can cause a cell to lose control and growth is by changing what we call the epigenome. So the epigenome is another code. We have the genetic code, which is the DNA, and the epigenetic code is the DNA and the proteins. So you have the DNA, which is always wrapped around these proteins, and this type of information together can be transmitted from one generation to the next. So it's not just the basic DNA code that can be changed or transmitted to from one generation to the next. You also have another level of information. And you can also cause cells to lose controls by viral infections. So in that case, a virus will get into a cell, and the virus will have a piece of DNA that will provide information to the cells to just keep on growing. So in order to explain what mutations are, of course, we have to consider what the genome is. So our genome, I'm going to work a lot by analogies, and I hope I found some good analogies. So our genome is like an encyclopedia. It's like an instruction booklet for the cells. And in our instruction booklet, so each of our cells have this instruction booklet. And each of our cells have this genome which consists of 3 billion letters. So these 3 billion letters, of course, if you were to print them into a book, into a series of books, you'd end up with these humongous books. So us researchers in our lab actually always consult the sequences of the genome through the internet. So all this information, whenever we do some research, is through these computer databases of human genomes. In cells, these 3 billion letters are organized into chromosomes. So in each cell, there's 23 pairs of chromosomes. You have one chromosome that comes from mom, one chromosome that comes from dad. And this is what is illustrated here in this panel. So here's a cell, for example, and this is where each chromosome is simply color-coded, so you can differentiate them. So from the cell, you have each chromosome. If we organize them, you can see this is the pair of chromosome number 1, 2, up until 22, and then you have here a female because there's 2 X's. So in each chromosomes, the letters are organized into words. It's just like in a regular book. You have these letters are organized into words, and these words, we call them genes. So in the genome, the genes are also organized into sentences that make sense. So for example, you can have 4 genes that are organized together to make something that makes sense. José is very sad. So I was asked, why did you pick that? But you'll see why I picked that. So in this particular case, José is very sad is 4 genes. It's a sentence and it makes sense. So we got the sequence of the human genome from a lot of people, a lot of different people. And what we found is that in the healthy population, so say in my genome versus in your genome, there's a lot of differences. So we said there's 3 billion letters into the human genome. Well, between one person to the next, there's millions of letters that are completely different, even though both people are healthy. This is what we call variants or polymorphisms. So this is an example here of a variant that does not change the meaning of the sentence. José is very sad. José is very sad. Or José is very sad. So here you have these changes, these polymorphisms that do not change the meaning of the sentence. And these polymorphisms are throughout all 23 chromosomes, and there are millions of them. So you say, well, how come if everybody's different, how come there's all these differences and you have so many healthy people? Well, some of these differences actually can cause changes in the meaning of our genetic code, and this is what we call mutations. So you have polymorphisms and you have mutations. So for example, here I put 4 of these types of differences between genomes that are mutations. So you have 1 letter changes that can change entirely the meaning of what genes do inside the cell. So for example, if you remove S, if you substitute it with M, so José is very sad, José is very mad, she's very bad. And sometimes you remove a letter, you change a letter, and the letter makes the whole sentence completely useless. You don't know what means José is very ad. So in the context of the whole genome, if you say José is very sad because she did not explain this properly, it's very different than if you say she's very bad because she did not explain it properly. So it's the same thing with insertions. You can have entire words that are introduced into sentences. Sometimes they're duplicated, and sometimes there's entire words that are removed. So if you remove, some words can be removed without much change to the meaning of a sentence, but sometimes if you remove a whole gene or a whole word, you're going to change completely the meaning of a sentence. So these mutations can occur everywhere in our genome, and they do not always lead to cancers. Sometimes these mutations actually can lead to other types of diseases like neurodegenerative diseases. So what kind of mutations actually are associated with cancers? There's 3 types of genes that if they are mutated are associated with cancers. you have the so-called oncogenes. So these genes in the cell, they promote growth of the cells. So some types of mutations will actually make these oncogenes more active. So in that case, you stimulate cell growth. There's also genes that will reduce the ability for a cell to grow. So if you have a mutation in that gene, you will no longer suppress the ability of a cell to grow, and it will start growing aberrantly. And then you also have mutations in genes that repair the DNA, which can lead to the development of cancer. So, for example, if you can't repair your DNA, you will acquire mutations through time, through generations. This is an example of a healthy genome. So to explain this or to illustrate this, I'm showing you the difference between a healthy karyotype and the karyotype of a cancer cell. So this is the karyotype you saw before. You have the chromosomes from mom and dad, and up until the sex chromosome. And in the cancer genome, you can see just by comparing these two chromosomes with these two chromosomes, you can see already there's a big difference. There's a piece that doesn't belong there because it wasn't repaired. And it's the same thing throughout the karyotype, throughout the genome. You can have the accumulation of more than 2 chromosomes. So this is what happens to your genome if you can't repair it. So I think one very important take-home message from my segment tonight is that cancer develops from numerous changes, from various sources. So you can develop cancer from genetic mutations, and it's never just one, it's multiple mutations. And these mutations can be either from already given from your mother and your father. And this will affect your susceptibility. So your heredity can affect your susceptibility to cancer. For example, here I put, this is my name. This is actually my husband's name. So if you look at my daughter, she acquired one set of genes from me and one set of genes from my husband. And then you can also acquire through your lifetime some mutations if you are exposed to certain types of chemicals and also to radiation. And this concept of cancer susceptibility will be discussed by Dr. Tonin tonight. And of course, we said that you can get cancer from numerous types of changes, not just genetic changes. We said also viruses can cause cancer, and epigenetic changes. So this topic will be covered by Dr. Yang. Okay. That's it.
During the time that, in fact, Dr. Yang comes in, if there's any questions, we have maybe 1 or 2 questions rapidly. If people want to ask José something about her daughter or about her work. So I think that's important to see that what we do in life is we cross many environments, and some of them will be much prone to promote these carcinogens, these mutations. And I think that's what the point that José wanted to make. You want to add a bit on this, or? Okay, so we'll pass now to Dr. Jinjiao Yang, which will talk to us about epigenetics. That's a difficult topic even for scientists, so we'll see how you can do, Jinjiao. Thank you.
Okay. Okay, my name is Xianjiao Yang. Some people would call me XJ. When I was in the US, people just call me by last name. I came from China, and I didn't realize my last name was something culturally. In China, actually, my last name was better. So in the culture, it's quite different among different countries. So what I'm going to do today, this evening, we'll be talking about just explain. Can you? What?
Yeah, we all have to learn the intricacy of speaking in these. I think this is good.
Okay. So I will explain what does this word mean, and try to relay some, things happen every day. And at the end of my talk, I will just try to, give you some ideas, what we can do to prevent the cancer. So one thing, since child I have been studying, doing research, and one question I ask myself, why I'm doing this. My friends, my family sometimes ask me to go for, business, things like that, or go to medical school. So there are two reasons. One is for my curiosity. Another reason is try to understand, biology, and then try to get some new knowledge. For example, so what is epigenetic? And epigenetic, this word is divided into two parts, epi and genetic. So epi, this prefix actually was initially taken from Greek, and it means near, on top, or above, but never the same. So therefore, the word epigenetic refers to heritable phenotypes that are not caused by mutations or changes in the DNA sequence itself, as Jose mentioned earlier. And this term was initially proposed actually for over 50 or 60 years ago, and only started to become popular in the middle 1990s, about 15 years ago. Actually, I learned this word at that time when I was a postdoc in the US. So epigenetic changes are reversible. On the other hand, genetic mutations cannot be easily reversed. So therefore, epigenetic change provide a good opportunity for drug intervention and also therapeutic strategies. And as Jose mentioned, that the genome, there's so many letters, and there are 46 chromosomes in a human cells. And if this DNA is really small, if you link them together and stretch out, actually, you will be amazed. It can be like 2 meters. It's quite long, a little bit higher than me. And the nucleus itself is very small, and actually where the DNA is put there. So the question is, how could that happen? And this process involves a compaction about 10,000-fold. So this is amazing. I'm sure everybody you do moving. For example, we moved the lab from another hospital to here last year. So moving, just a few things take a lot of time, and it's very difficult. And these compactions, how could that occur? So this involves a delicate packing and unpacking system. So you have the DNA, this is the double-stranded DNA. So this DNA there. So this is the letters that Jose mentioned to you just a few minutes ago, contain all the genetic information. So then those DNA interact with proteins called histones. Those are proteins, and form this round structure that's like a bead. And those link together, so then form like a string, and then further folded to form the chromosome. So we learned a lot about this step, but really little is known about this step. So those still a black box. And this is the packing process. So, when you move things from move a lab or move apartment, you pack things. So then next question is unpack them. Sometimes you move apartment, leave things there maybe for several months, still couldn't get it back. So same thing here. So basically, once you pack during after cell division, this chromosome has to be unpacked. So this actually involves a different marking system. For example, add like chemical marks there. So different marks on the DNA and also on the histone proteins. So different types. And this mark controls which region is more tightly packed than the others, and which region is unpacked first after, for example, after moving your apartment, this box should be unpacked first, delivered to that bedroom, for example, things like that. And those are chemical tags. Those tags form basically another layer of information on top of the DNA sequences, on top of the genetic code. So this layer of information we refer to this layer of information, something called epigenome. This is a very new term, just came out a few years ago. So this is even more complicated than the genome itself. Moreover, each cell type, for example, in our bodies, there are so many cell types. Each cell type has its own distinct epigenome. And Jose mentioned earlier about her daughter, for example, from development after birth. So this is a fertilized egg during development give rise, a fetus and then an adult after 40 or 50 years later. So in him or me, so there are many cell types, as I mentioned, muscle, like in the brain, there are neurons, and on skin cells, there are different cell types. But all those cell types have the same genome. However, their epigenomes are different. Same genome, but distinct epigenomes in different cell type. This actually, I was preparing this talk yesterday, try to make it good. So afterwards, I watched a movie. That actually was quite interesting. So some of you may watch this movie. How many have you watched this one? So this seems quite a popular one. So initially, this is actually a very interesting movie. So this is basically, you can reverse this process. I'm not sure this really can happen in real life. However, now scientists can make one cell at least. For example, a muscle cell go back to the stem cell. And in this case, actually, involves you need to remove all those chemical labels. Okay, so this is fictional. I don't know what happens in the future. So this is another case here. So you have 3 identical twins, genetically identical. Okay, so this those ex-movie stars, and I'm sure you know this pair. They actually were very famous 200 years ago. They are co-joined. They almost co-joined similar environment, but they were like complete different individual, different personality. However, their genomes are the same. So, for example, I'm not sure which one. I heard one of them really was cheerful and didn't enjoy alcohol too much. The other one was less cheerful and was alcoholic. You see the same genome and different personalities. And also, as I should mention here, you have the girl, and each girl has 2 extra chromosomes. You know the chromosome Jose mentioned, the X chromosome? One girl has 2 copies. For me, for example, I have 1. So then there's something called X chromosome inactivation. Only one of the X chromosome in girls or ladies is active, and the other one is inactive. Why? Because one of them is tightly packed, and the other one is more loosely packed, and they contain completely different chemical marks or epigenomes. And this also happens in this cat. This is called a lucky cat. Actually, you have the different fur color here. This dark patch, and this is brown or yellow, or the white patch. This is because of distinct X chromosome inactivation. And the white patch doesn't contain any pigmentation cells. The dark patch, for example, the X chromosome that contains the black pigmentation gene is inactive. And the other one was inactive. And this is reversed in the brown or yellow patch. So this also gives you an idea, Some of you must know this cat, this type of cat. And so this is basically tell you that they are epigenetic marks. And those marks control the gene, which gene is active and which gene is inactive, and when and where. And this is very important. So if I have time, I will just show you a very short video from YouTube, 1 minute. Just rephrase what I just said. This one? See, this is a DNA. I don't feel the sound. So this is the histone protein, the DNA wrap around. So now the chemical tag is added there. Okay, it's just like what I said. This is the epigenome. And then they link together. Okay, so then a lot of them. Okay, not just one or two, many of them. And this determines the structure of the genome and how tightly packed and unpacked. Okay, so then each different cells have different, epigenomes. So these are different cells, you see. Okay. So then if those marks are wrong, so then this will cause mutation. So that could be the reason for cancer and other diseases. And then question is how to use this information to prevent disease. So for me, myself, that's a question I mentioned earlier, how we can use the knowledge to prevent something from happening before it started. So there are actually now, we don't know a lot. It turned out that, for example, what food you take and also how much you take can affect the epigenome. For example, red wine, red grape, and also fiber-rich food. For example, in Asia, fiber-rich food, people tend to eat more like vegetable, fiber-rich food, and colon cancer risk incidence is much lower there. So one reason is because of there's a compound that can maintain the epigenome correctly. And also, lifestyle and environment has a huge impact on the epigenome. For example, exercise, smoking either positively or negatively affects the epigenome. And social interaction, parental care, and may also affect the epigenome. And finally, physical environment. So basically, to summarize what I said, the genome is relatively fixed in life. However, the epigenome is much more dynamic, may change according to the environment. And then wrong epigenetic marks can cause cancer and other diseases. And healthy food and active living styles may be good ways to maintain the epigenome correctly and to prevent epimutation from happening. And we don't know a lot, so therefore we need do more research. So finally, I would tell you that take care of your epigenome, stay healthy. Thank you.
But there's one good message out of this, is that if the epigenome is bad, so you would think that you could, in fact, improve.
Yes.
So you're not there and condemned to have mutations and
yes.
cancer. That's
I think the last speaker, Daniel, will tell you there's drugs actually can collect the epigenome.
Use the mic. Can you put just to let you know also, in the past, I think 3 years, there are now compounds in the clinic to treat certain cancer by modifying the epigenome. The acetylase, the enzymes that Jin-Jean is working on, has been used now in clinic and seems to be extremely good. We will talk about that a bit in some leukemia and lymphomas. So even it looks a bit, very basic, very scientific. This knowledge allows us to move forward to new treatments.
Okay. Can you hear me? No? Yes? Good. Okay. This better? Okay. Okay, so I will talk to you about the genes that are involved in cancer. Genes or proteins, okay, that cause cancer. So as you know from the previous speakers, there are a lot of them in our genome, a lot of genes, and only some of them are involved in cancer. Now to find them is not easy, okay? So I'll give you an example of a disease that is terrible for the people that have it. It's wonderful for us because we can find the gene, okay? And a lot of these cancer-causing genes were found through these hereditary tumor diseases, okay? So these are people that have a acquired mutation, so they have a mutation that they get from their mom or their dad, and that makes them prone to develop cancer in different organs, okay? Because they already have one mutation inherited. Okay, so I'm going to talk about a disease that's called the von Hippel-Lindau disease, very long name, or short VHL, and how this discovery of this disease and the disease gene led to a treatment of kidney cancer. Okay, but first I have to explain to you what these cancer-causing genes are. Okay, so they're divided into two. One is called oncogenes, and the other one is called tumor suppressor genes. Oncogenes are very simple. So this is a cell. These are two chromosomes, one from mom, one from dad. You get a mutational event. So a mutation happens that now changes something in this oncogene. And what happens? This oncogene is now hyperactive, and it causes cells to proliferate abnormally. So normally cells would just not do anything, like most of the cells in our body are not proliferating. However, when you get a mutation like that in an oncogene, this gene now starts to act where it shouldn't act, and cells proliferate. Okay, so all you need is one mutation here. Then you have the second class, which is called tumor suppressor genes. So they do exactly the opposite. They stop cell proliferation. And when they are gone, when they're inactivated, when they're removed, then the cell can proliferate and divide. So in this case, here again you have your two chromosomes. You need to inactivate these tumor suppressor genes twice because you have one copy from mom and one copy from dad. And just getting rid of one copy is not enough. You have to get rid of both. And so it's more rare that this happens because you need to get rid of both of these copies in the same gene and in the same cell. Once that happens, you have two inactivating mutations. Now this gene is no longer functional, can no longer suppress cancer or tumor formation, and now you get cells that proliferate abnormally. Now the disease I'm going to talk to you about, and there's many diseases like that. They're usually very rare, and that's why most people don't know about them. In this disease, people are born like this because they already got one inactivating mutation from mom or from dad. That means they only have to lose the other one to initiate a cancer or a tumor. So they are much more likely to develop cancer than people like us who look like this. They have two healthy copies. So these people have usually quite a horrible life in that they continuously develop tumors in different organs. Okay? And it starts sometimes from very early on, like 2 to 3 years old. And it follows them their whole life. Now, this was horrible in the early days. Now it's much better because you can sequence these things and you can find out if a physician sees that somebody comes with a specific tumor, they can now sequence and see, okay, this person has this disease, and they can then screen these people by various techniques, X-ray, MRI, CAT scan, to basically take out the tumors before they cause any harm, before they metastasize. Okay, this is just another quick example of what these oncogenes, for instance, can look like. So these in red, you see the chromosomes of a cancer cell. And this would be the normal case. So the oncogene has 2 copies, and you see 2 yellow dots. So this is in a normal cell. Now this cancer cell, you can see already that there's many chromosomes that have 2 red dots. Some have 1. So there's already something happened here. So there's a lot more than there should be. And then you see this chromosome, which has probably 1,000 dots. So this oncogene has been duplicated or has been multiplied 1,000 times. And so the levels of this protein in the cell has increased 1,000 times. And that drives this cell into a tumor cell. Now, a tumor suppressor gene will be much harder to detect because it's actually gone in a cancer cell. And then you can't see it anymore. Okay. This is how we study cancer in the lab. This is one way. We do cell culture. So we have a petri dish shown here. We put normal cells in here. They're usually from rodents, mouse, or rat cells. And we then overlay that with a media that makes these cells proliferate. And these cells proliferate only until they fill this first layer, and then they stop because they touch each other, and they are contact inhibited, and that's a signal for them to say stop. Just like the cells on your skin, they will stop at one point. They will not continue to proliferate. Now, if you introduce a gene, like an oncogene, into these, into these cells, that's what happens. These cells now do not stop to proliferate. They pile up, and they form a tumor-like growth in cell culture. The same thing happens if you would inactivate 2 copies of a tumor suppressor gene in this cell. Same thing would happen. And you can measure this in the lab, and that's how we classify these genes and test them. There's many other tests. You can go into mice and do more sophisticated tests. Okay, so now to this disease. It's called the von Hippel-Lindau disease. Why? Because 2 physicians in a very long time ago, about 100 years ago, discovered this disease. What they saw is that the patients that have this disease often have tumors in their eye, in their retina, that bleed, and these people become blind because their eyes are full with blood. They also saw that these patients often have, after the patients died, have benign brain tumors, spinal cord tumors, and also tumors in the kidney, as well as cysts, which is a thing that goes around the tumor, a fluid-filled vesicle. Okay, so this is the disease, and you can see it's quite bad. You can get up to 500 tumors in a lifetime. This disease is now treatable in a sense that when you have the disease gene, and the physician knows that, you can take these tumors out before they become too dangerous. And so people with this disease now live into their 60s. It's not a fun life, but it's better than no life. Okay. The gene for this was discovered about 100 years later, 1993. And it took these people, 30 scientists in the US and other places around the world, 10 years to find this gene. What they did is they compared the DNA from 200 different families around the world, and they checked which gene is mutated commonly in all of these families. And they found this gene. It has a certain amount of amino acids, and these are the mutations. And in red, you see the mutations that were discovered in the tumors from the people that have this disease. And in green, this was a surprise, you have the mutations that were discovered in people like you and me that don't have this disease, but that come up with kidney cancer at one point in their life. Kidney cancer is usually caused by smoking, and so people in the 50s or 60s often develop kidney cancer. 4,500 people in Canada per year. And now the interesting observation was that the people with kidney cancer have the same mutations in the same gene as the people with this disease. So it's basically the same gene that causes both types of diseases, and you can now study this disease, which is much more frequent than this one. Okay, so this is how people think kidney cancer develops. So we're talking about the kidney. The kidney is lots of tubules. And what do these tubules do? They filter the blood and make it into urine. So this is a normal tubule. You have maybe one VHL mutation, if you're born with it, and nothing happens. Now, the second mutation, inactivates the second copy, and you now get a cyst. So these cells now start proliferating until they become a much bigger tubule. This bigger tubule probably doesn't work properly anymore, but since it's local, it will not cause any harm. Now you need a third hit or a third mutation, potentially in another oncogene, to make this cyst into a tumor, which is then called renal cell carcinoma. And then you potentially need another 2 or 3 or 4 or 5 mutations in some other genes to make this tumor into a tumor that spreads to other organs, which then becomes really dangerous. So in this case, you can easily remove the tumor from the kidney, or can you remove the entire kidney? That's not a big deal. You have no symptoms, but when it spreads, it becomes very dangerous. Okay, so this is what we do in the lab. And this, I'm not going to explain this to you in detail, but it took then another 10 years, from 1993 to 2003, of basic research, and this is what we do in the Cancer Center, in many labs around the world to unravel the VHL pathway. So we started with this gene. We know the disease is caused by the inactivation of this gene. So it's removed, it's no longer there. But it doesn't really help us because we can't make a drug against something that's not there. So we have to make a drug against something that's there. And so this gene, when it's gone, leads to the accumulation of this gene, which you could call an oncogene. So there's too much of this gene called HIF. And this, in turn, turns on 3 different oncogenes that are involved in either cell division or blood vessel growth. And luckily, for the people with kidney cancer and the people with the VHL disease, there were certain pharmaceutical companies during that period that were making drugs against these different oncogenes because they're also involved in other types of cancer, breast cancer, lung cancer. And so these drugs were then available to try testing them on people with kidney cancer. And this is all based on the research. This wasn't just a random trial. And so this is an example. So as I said, regular kidney cancer is not dangerous because it's localized to the kidney. If you detect it early, you can remove it. When it's metastatic, there was nothing that could be done for those patients. They survived a few months, and there was no treatment because it wasn't responsive to any chemotherapy. Now, the treatment with this of the metastatic kidney cancer with drugs that were discovered through basic research led to a tumor shrinkage and a doubling in survival time. So these people eventually still died of kidney cancer, but they lived twice as long as before, which sometimes can be years. And so that was a big breakthrough because before there was nothing. And so here's just an illustration. This is a metastasis in the lung, and after treatment it's gone. This is basically, here's the one kidney which is still healthy. This kidney was removed because there were a lot of tumors in there. And you see there's cancer here. There's another metastasis here. And after treatment, they're down to almost nothing. So that was my message for you.
Well, I think you better give us a more happy message than this. But the point is, thank you very much, Art. So before we go for lunch, just so your message is what? Is it the positive things is that there are many nice positive research. There's a question there, and this is moving. Please. Can you repeat the question?
Not every gene is an oncogene, but
No.
just one gene is an oncogene.
No, there's many.
What percentage of genes have the capacity to be an
What
oncogene?
percentage of genes have the capacity to be an oncogene?
I mean,
Probably less than 1%.
that's a discrete thought. Like
Yeah.
Not just one gene is an oncogene.
No, it's maybe
But not
maybe
just
100, maybe
one
50.
gene is an oncogene.
No. Very, very few. And they all cause different diseases. No, the message from this talk is basically that basic research and understanding different diseases in cancer takes a long time and takes years, takes decades. In this case, it took over 100 years. But in the end, you find something that helps people with the disease. This is not perfect. This is no cure, but it's a start. And it has taken people 30 or 40 years of doing nothing, where people just basically said, okay, this is the disease and you have that much to live, to now an option.
Well, I think there's another question. Yeah, there's one in the back.
You basically follow with MRIs, and you see what happens. I mean, these tumors are pretty easy to detect, because these types of tumors, because they're highly vascular, a lot of blood vessels in them, so you can see them very easily. With other tumors, it might be more difficult.
I think we can continue. We'll have the questions after when come to ask Arnie. I think we want to move on and have there's a nice cocktail, some food upstairs. And we'll take about 10-15 minutes for break, and we can come back. And then we'll talk about more what Denis and Pat has to say about this. Thank you very much.